Good afternoon, everyone. My name is David McHugh. I'm the Associate Dean for Research in the School of Agriculture and Food Science. And we're delighted to welcome you all here today for the first seminar of 2021 in our research seminar series. I'm going to now hand over to Dr. Emma Feeney, who will introduce today's speaker and chair the session.
Thanks very much, David, and welcome everyone who's joined us this afternoon. It's my great pleasure to introduce Marco. Marco completed his PhD in Spain in 2011 and then joined us here in UCD as an assistant professor in food science and nutrition in 2020. Marco's primary research interest focuses on the utilisation of microalgae as foods and macroalgae, and his research explores the use of extraction procedures with valuable compounds using innovative technologies for their use as functional foods, as well as the characterisation, biological properties and safety aspects of their use. So this afternoon, Marco is going to tell us about some of his research on seafood from the sea to your table.
Thank you very much, David and Emma, for this introduction. And thank you so much to the school for inviting me to present here today, especially in the first seminar in 2021. And I'm just going to talk briefly in these next 25 minutes about a few of my research experience working with exploring functional foods from seaweed, what I entitled here, From the Sea to the Table. Just thinking backwards on when my interest in seaweed started, I can say that my interest started in 2011. Once I finished my PhD, I was exploring the influence of different mineral supplements in animal production and also animal health. And one company from seaweed was interested in developing actually one mineral supplement using seaweed. However, my PhD was over and I moved to industry for a couple of years, 2-3 years. Working in the dairy industry, seaweed continued following me, and I was in the research and development program, and they asked me to develop a cheese containing seaweed. Then later on, moving on, I went to a pharmaceutical company in marketing, and I was also having in my catalog drugs from seaweed. So finally in 2014, I came back to research with my own postdoc, and I started in Chagas doing some research about this mixture of food and pharmaceuticals compounds that can be extracted from seaweed. Continue as a postdoc also in UCD since 2016 in John O'Doherty and Torres Sunis group. And in 2019, I joined Chagas as a research officer in charge of all the marine research in Ashton. And in 2020, I joined as an Astra here at the school. So as you can see, I have a strong interest in this biomass. And for those of you who are not interested, maybe this will catch your eye. These are data from the Food and World Organization in 2014 that estimates that the seaweed produced in the world is valued in approximately 8 billion per year. All these correspond to a biomass production of 25 megatons per year. And from this biomass, we can see that the main producers currently are China, approximately 62%, other Asian countries 37%, and only 0.2% correspond to other parts of the world. For those of you who are not really familiar with this crop, we can see that there are multiple species and they can be classified based on their pigments in 3 main families. They are brown, that are really common here in Ireland, green and red seaweed species that we have also quite a few species here. From brown seaweed, there's approximately 2,000 species. Their main pigments are xanthophylls. They are really variable in size from 1 to more than 100 meters, depends on the location. And they can be growing in different areas all over the littoral zone. And they are mostly marine. In the case of green seaweed, there are more than 1,000 species, and they are green because their main pigments are chlorophylls. They are shorter in size, from 1 to 2 centimeters to 1 meter, and they can be also present not only in the sea, but also in fresh water. And red seaweeds, that are also really common here in Ireland, and they have many common uses. They have more than 7,000 species, Their main pigments are phycoerythrin and phycocyanin that confers this red pigmentation. They can be up to 60 meters, and they're also mainly marine. So as you can see here, when I talk about seaweed, we start to talk about variation, and that's one of the words that are going to come up in this presentation a lot. This variation is also reflected in the composition. The only constant thing that I could think about seaweed is that they are really rich in water. So independently of the species, most of them are going to be composed of 80 to 85% water. And once you take that water out, it has a really rich mineral content, approximately 30% of ash. And then everything is variable from there, depending on the species, the location where you take the seaweed from, and the season of collection. Once the seaweed is dried, we can say that they are really low in fats, between 0.5 to 7%. Rich in protein from 5 to 35, described in the literature, and mainly really rich in carbohydrates from 40 to 65%, and from those carbohydrates, really rich in fibers. If we focus not only on the amount of these nutrients, but also in particular compounds that are really nutritious and not really relevant in our diets, we can see that bioactive compounds from seaweed that can be found in high amounts are polyphenols, fatty acids, mainly polyunsaturated fatty acids that could be included in the diet, omega-3 and 6, carotenoids, sterols, and the main topic of this presentation, which are going to be proteins and amino acids, essential and non-essential amino acids, and polysaccharides such as fucoid and laminarin, but also others such as carrageenan, ulban, and alginates. And I mention all these particular compounds because they have been found multiple uses, not only in nutrition, but because they are also variable depending on the species, and they can have many biological functions, including antioxidant, antimicrobial, and anti-cancer. So the main question is why are we not including seaweed in our diets more often? And as I mentioned before, it's mainly because of the ash content. So once you dry it, 30% are minerals, and that's not really good from a nutritional point of view. So what we're currently exploring is extracting these elements that have biological properties from seaweed. The general process of extraction goes as follows. Basically, you have the full biomass and you're going to process it, you're going to dry it, you're going to mill it. And then this biomass, you're going to mix it with solvents, water, acids, or ethanol. And you can use here also to increase the yields of compounds that you're taking out of the seaweed, other technologies, ultrasound, high pressure, microwave. And at this step, seaweed stops being relevant. You take out all the seaweed biomass and you end up with a fluid because you're mixing it with solvents. And these are called crude extracts. At this step, you also measure the biological properties, antioxidant, antimicrobial. And if there's something interesting there, you can continue with some purification strategies. You can use solvents, you can use membranes, and also chromatographic techniques, but just to be sure that you use them in low amount for lower period of time, because these purification techniques are really expensive. Once the compounds of use are really purified, you can start thinking about industrial applications, and in this case, I'm going to focus on nutraceuticals and also food supplements. So once we have an overview of the research that I'm going to explain in the area, I'm just going to focus mainly in 3 different research experiences that I have here as part of the school and also as part of Chagas in the extraction of these compounds. And then once they are extracted, how are we going to use it as part of food? In the case of extraction, there have been multiple studies exploring the extraction of different compounds. And originally, the study started by doing what we call a screening testing. What it means is that people were exploring seaweeds and were using different seaweeds with different technologies to obtain crude extracts. They were using other seaweeds with the same technologies to obtain multiple extracts, and all of them were tested for their biological activity. And they obtained really great results in biological testing in vitro, cellular assays, microbial assays, and also animal trials. Really great results. However, the most important thing is once you want to make some claims about the food or a feed, you have to go back to what is the active compound. And since all this was a screening, once you check back at the composition of these crude extracts, you can actually check that this composition is really variable. So there's not really a compound that you can think that is the main factor influencing this bioactivity. Due to these multiple problems, the next generation of extraction studies, which is the ones that I'm going to focus on today, are called target testing. So you work together with an industry or somebody that is interested in an industrial product. In this case, you focus on the final product. What is your desired product look like? Imagine that you have a final product like here on the right side, and you're focusing on this product to have really rich content in polysaccharides. Based on the previous screening literature and using those technologies, you can use the protocols available. And once you explore this, we can check that the technology that works best to obtain, in this case, this desired final product will be these two technologies in the middle in this scenario. And like every technology that you use in the lab, it can be optimized. There's many processes that you can optimize. One of the most common ones that everybody understands is temperature. You can use every technology that you can think of at different temperatures. In this case, I selected low, medium, and high temperature, and you apply these processes on both technologies. And when you do this, we can check that in this scenario, this technology at medium temperature will be the most appropriate to obtain a crude extract that looks really, really similar to your desired final product. In practice, this will mean that you're saving money because you will not be using a lot of purification strategies. You will save time doing all these extraction procedures. And at this stage, all the in vitro assays will be more repeatable. You will have better results and more homogeneous. Once we understand more or less where this optimization, I'm just going to work through one optimization work that I did of one technology, ultrasonic technology, to recover polysaccharides, in this case from brown seaweed, and antioxidant activities. This was funded by Science Foundation Ireland, and you have here the full publication in Marine Drugs in case you want to. It's open access in case you want to read more about it. In this case, we're interested in two polysaccharides from seaweed. So if you take into account the full biomass that you can amplify to a cellular level, the first polysaccharides that we're going to be interested in for this study are laminarin, which are some reserved polysaccharides that are in the cytoplasm of the cells. And if you could amplify a tiny bit more into the cell wall of seaweed, you can see the second polysaccharide of interest, fucoidan, mixed with other compounds present on the cell walls of seaweed. In this case, we're going to be interested only in these two, extracting these two, and monitor at the same time antioxidant properties, although we are aware also that these polysaccharides can have multiple activities. The technology of choice in this case is ultrasound-assisted extraction technology. For those of you that are not familiar with it, this is an ultrasonic probe, and once in a liquid medium, these ultrasonic waves travel through the liquid medium, generating bubbles that are going to grow and explode and implode, also generating these forces of extraction. So the main objective is to actually optimize all the process parameters that we can control with this technology to obtain high amounts of this polysaccharide. As I mentioned before in the previous scheme of the general concepts of extraction, we process the biomass. And this is how ultrasonic extraction probe looks like in real life. For this particular experiment, we are going to control 3 different extraction variables. We are going to control the temperature from 40 to 80 degrees, the time of extraction, 10 to 30 minutes, and the amplitude of the sonication waves for 40 to 100% of sonication waves. And we are going to do this using response surface methodology. What that means is that we have to do a full experimental design, making different combinations of temperature, time, and amplitude. And once we do this and analyze our results, we can check already few patterns here in the data. In general, the amounts of fucoidan that we were obtaining at really higher temperatures within the range, higher time of extraction, and medium amplitude. In the case of laminarin, really lower conditions of almost all the process parameters. And antioxidant activities, we could see kind of medium between all of them. All this data, we have to model it. And I'm not going to go into much detail about how model is done. I just want to explain why these graphics are important. So this is called response or phase methodology graphics. And if we can just amplify into one of them, we have three different variables. If one of them, the ultrasonic amplitude in this case, that is here at the top, maintain it constant, you can check the influence of the other two variables, the extraction time and temperature, on the amount of antioxidant activities that you have. So in this particular case, you can see that the antioxidant activity is especially high in the middle, so 25 minutes of extraction time and approximately 70 degrees of temperature. All these graphics together for all these compounds were taken into account when designing the optimum extraction conditions that I record here. We created for this technology 4 extraction conditions that were meant to be optimum. One for fucoidant, one for laminaring, the third one for antioxidant activity, and the fourth one for all of the compounds together. And the software says that if this model is correct, applying these extraction conditions, you should obtain in the final extracts a concentration that is within these ranges. So we repeat these extraction conditions, and we could check that in general the experimental results were really well with those predicted by the model, except in the case of antioxidant activities that were a bit tricky, especially within the lower limit. So what does this all mean? Main conclusions are that this ultrasound technology can be used to obtain these compounds, and we can optimize the conditions to obtain the compounds of interest. And the main impact for industry is that when we were using a traditional protocol with the same seaweed and extracting it the way that the industry was currently doing, we were obtaining in those extracts approximately 0.1% of laminaring in this case. And with our optimized protocol, we were obtaining 0.7. It doesn't look like much, but this is an increase of 7-fold. And the most important result is that if we compare the time of extraction with the traditional protocol, we were using 32 hours of time, and with the optimized protocol, 10 minutes. Other way to explore technologies, which is a bit more advanced, is exploring the combination of two technologies or two forces altogether. In this case, we explore ultrasounds, microwave, and the combination of both. And this work was performed in Chagas, also funded by SFI. You can find here also in open access the publication in case you want to explore it a little bit further. Ultrasound technology, as explained before, the main mechanisms of how to use it. In this case, the only difference is that we designed this vessel containing the sample. Microwave-assisted extraction, we apply the same vessel inside a microwave system. In this case, microwave works in a different way. So microwave heating, what it does is it heats the molecule inside the cells. So those molecules start to vibrate because they are really energized. They start to vibrate and they start to disrupt the cells. And these cells, once they are disrupted, they release all these compounds outside the cell to the media. So in this case, the heat comes from inside the cell to outside, and so following the same direction as the solvents. And for this case, we also design in the lab our own apparatus to perform the extraction. So to perform the combination of both, we can also do it in the microwave in the same system that we designed in that vessel, and also applying the ultrasounds all together. The main, this is the full experimental design. So we were mainly interested in exploring different combinations. In this case, ultrasounds, as I mentioned before, different amplitudes, similar as in the previous case, between 20 and 100%. Microwave, different also microwave powers between 250 and 1000. And the combinations of both, all the possible combinations of sonication amplitudes and power during only 2 and 5 minutes. The main interest in this case was to analyze, again, fucoidant, but also total soluble sugars, phenolic compounds, and antioxidant activities. But just for this session, I'm going to focus on the same polysaccharide fucoidant that we covered before. Taking a look at the results, when you use only ultrasounds within this system, we can see that the ultrasonic amplitude had a strong influence on the amount of compounds that we were obtaining. Also time, 5 minutes always obtain more than with 2. And the maximum amount of compounds were approximately 200 milligrams of fucoidant per 100 grams of seaweed. Huge improvements were also seen when you applied microwave technologies. In this case, also microwave power have a huge influence, and also the time of extraction. You can see here that microwave applied for 5 minutes and 1000 power obtain approximately 9-fold more than the previous technology. And the most interesting results were when we combine both forces of extraction simultaneously, so both of them together, we can see a huge influence of time as well on both parameters. The most important part is that now we are obtaining approximately almost 4 grams per 100 grams of seaweed by combining, and in only 5 minutes. All these results, we can also see it in the seaweed. So to do that, to see if there was an actual influence on the seaweed, we can check the microscopic images. This is how the full undried seaweed biomass looks like at the microscope. You can see that the surface is smooth and it has a kind of impurities attached in the image number B. And below you can see that it has many impurities attached. Those are actually salt residues. Even though you are trying to clean the seaweed, there's always some salt residues present there. Once we apply microwave extraction for 2 minutes, which was the one that achieved the lowest yields, we can check that this surface is not so smooth anymore. It's a bit more disrupted. And in the image number B, there's way less impurities. And with the condition obtaining the maximum yields of compounds, we can see that the biomass is totally disrupted. You can see even the formation of different channels, which is the ones that are going to be leaking the compounds outside of this biomass to our extracts. So in this case, we can see that all of these technologies, but also both of them applied simultaneously, have really strong abilities, or they can be really useful in industry when we're extracting these compounds. And the main impact for industry is, if you imagine that in the previous one we were obtaining already the compounds in 10 minutes, in this one we're obtaining these compounds at the same levels during 2 and also 5 minutes. So this it will imply for industry a huge improvement, both in the time of operation, the time that people need to actually achieve those compounds, and also the energy that they are going to use is way less. And once we have all these compounds extracted, I'm also going to explain one example of how you can incorporate them into food, or at least check if they can be incorporated into food. So this work, in this case, was done with proteins, and are we going to incorporate seaweed proteins inside a food matrix? This was done in Chagas and was funded also at the later stages of Nutritional. You can have also the publication in open access in case you want to check it. In this case, we were working with a sonication procedure. This is basically, this protocol was provided by industry. So we obtain a crude extract, look like this. And then following the same industrial procedures that we were recommended, we purified with solvent twice. And this is the extract the industry was interested in using in a food matrix. This extract is made of 85% of proteins and 15% of other impurities. And our main question is, how, first, is it really useful to include it into a food product? Why are you going to include it? And second is, how are you going to do that? What type of products can be designed with this protein ingredient? So the first results, once you have a protein ingredient, the first question is, what type of proteins are actually present there? So we simply run a gel and we see that the proteins were actually low molecular weight, in this case, in this protein extract, between 71 and 27 kilodaltons. And question of why to include it in a food matrix, is it really relevant? We have to go back to the composition. So we analyze the amount of essential and non-essential amino acids of this extract. And when we analyze the data, we can see that the amount of essential amino acids was approximately 47% of the total amount of the extract, which is really high and is even higher than proteins from terrestrial plants that we commonly use in foods such as soya. So once we are interested in using it because of this composition and it's nutritionally relevant, the question of how are we going to use it or in what type of products comes from the properties of this protein. For this, we analyze the foaming properties, which is the amount of foam that we can create using this protein, and for how long is this foam going to be maintained in a food product. So we can see that it's going to be, depending on the pH, between 6 and 8, which is the most common foods, it's going to have really good foaming capacity. And the foam is going to be really stable, if you see here in the graph on the right, approximately 15-30 minutes, depending on the pH. So we can create huge amount of foam, and it's also quite stable. And the other activity is emulsifying properties, or what it means is when you mix this protein with an oil, the amount of emulsion, or the amount of mayo, if you want to call it, that is created using this product. We can see that it also depends on the pH, but also on the type of oil that you use. It has really strong emulsifying capacity. So in the common pHs for food, 6 and 8, we have minimum of 80% of emulsifying capacity, meaning 80% of that protein is going to be making emulsions. And the emulsion stability, if you heat that mayo that you have created and that you're spreading into your sandwich, how much of that mayo is not going to be transformed into a fluid? So that's checked by emulsion stability. And we can check here that depends strongly on the type of oil that we use. So between pHs 6 and 8 is really stable as well. And it's mainly stable if you use sunflower, olive, and peanut oil. These results are really relevant for industry because we obtain really similar results to those by many other researchers using egg white, which is a commonly used food, it's commonly used in pasta, it's commonly used in dippings and also in different pasty products. So the main impact of this work is that we can use this protein to develop vegan products. There are a common trend has been being at consumer trend since I think 2018 and continue to increase this consumer market. And finally, I just want to thank first the collaborations that I have in UCD and Chagas, and also the funding received to perform this work from Science Foundation Ireland and the BioOrbic Research Center, these two different grants, Nutramara, and also the Fundación Barrier, which were the ones that brought me here to Ireland to start these research projects. Thank you so much for your attention. I'm always open for collaboration if some people is interested in this work. So you can contact me here. You have all my details. And of course, if you have any question, I'm here to solve them. Thank you so much.
Thank you so much, Marco, for a really interesting talk there. I'd like to open the floor up for any questions. So folks, if you do have any burning questions, please pop them into the chat box there. Marco, while we're waiting for the questions to come in, might just have a few of my own. You've been working with a lot of different technologies there. Which one do you think is the best technology for some of the seaweed bioactive compounds?
So in my opinion, as you said, I touch many of them. And for me, the ones that actually can be combined with others are the most promising ones, because you don't have to use them in isolation. So once this combination of technologies can be achieved, I think that's the best way to go with technologies. Even if you have a conventional technology and you're applying an innovative one to improve a process that is already been demonstrated that it works, I think it's a great approach to follow.
Okay, great. And then in terms of the actual compounds, which ones do you think we should really focus on? Are there any that you've come across that you feel are more promising when you've been studying these different seaweeds?
So seaweeds are really variable. So for me, they are really interesting because they are really variable in composition. Every single time that you touch them is something totally different. So it's great to study biological properties. And in my point of view, I think the industry is moving towards sustainability. Mm-hmm. So I think in that sense, the sequential study of all of them to obtain a biomass that you can extract things from the biomass, but nothing is going to waste. I think that will be the ideal approach when it comes to seaweed.
Absolutely. And then in terms of the challenges, Marco, working with seaweeds, what would you feel are the main challenges there that you come across?
First, depends on the final use of the compounds. So if you're thinking in an industry, the main thing will be where is the biomass coming from? So that's the other area of research of biology. And then the second one will be the variability of these compounds. So if it's something really specific or if it's a specific claim, it will be really important as well to engage with the legislation side of the research. So contact EFSA in advance. If you're interested in making any claim, make sure what type of claim you're interested in from the beginning of the project, from the legal perspective, I think.
Okay, great. Thank you. So some questions are coming in the chat as well. And I think this is one that a lot of people are wondering about. Are there any concerns about the taste of the seaweeds? For example, if you're intending on adding these to a food as a food ingredient, or are there ways that you can circumvent this through the processing?
So the first thing is the taste is only relevant if you are using properly full seaweed itself. The same way that we perceive that umami flavoring compounds are mainly in seaweed itself, then polysaccharides are similar to terrestrial plants, really.
Okay.
Proteins as well can have an umami taste, but then depends on the amount of those compounds that you're going to incorporate into it. So if we think in a mayo and you're using olive oil with it, if you have normally the percentage of protein that you have there is 3%, highly doubt there is going to be much difference in consumer perception. But it's also a really interesting research topic and definitely has to be explored more.
Yeah, and it seems to be one that sort of springs to mind for a lot of people. I suppose on that same line of thought, is there a perception, do you think, by consumers that even though, as you say, the actual protein content for some of these ingredients might be very low, is there a perception by consumers that if it's a seaweed extract, it will have that sort of marine sensory quality to it?
So for what based on studies, I've done one actually this year that was published in microalgae in this case in Spain. So most of the consumers, in the case of microalgae, they just didn't know what it was. So that's one thing, or they were not aware of any benefits. And then in terms of the flavoring, I think the perception in most of the studies, what people is saying, that the people perceive it as really green ingredient and really nice. But as you're saying, really strong marine flavoring. So it's just about of daring to include it into different products, because there are many products that we consume that we don't even realize that they are made of seaweed until we check the label.
Okay. Okay, and some other questions then have come in the chat. Additional safety issues, perhaps, so around the content of iodine and arsenic that might be quite high in some of these seaweeds. Are these extracts depleted in the minerals, or could you comment on that at all?
Yeah, so in general, for what I can see as well, because I studied the full biomass and also the extracts, and I was also involved with the Food and Safety Authority in the latest report related to this. So there's a lack of information in many of the compounds because seaweed is really variable. So in the full biomass, it's true, they are really rich in minerals, and we have to be aware of especially few of them, like arsenic. But then you have to take into account that the amount that you consume of seaweed is really low. So that's one thing. And then the amount of that mineral that is present in the seaweed is not fully available. There are many studies that say that the amount of arsenic, which is one that people is actually thinking most of the times, like toxic arsenic, and that amount is less than 70% degrades after the digestion process. So it's 30% less viability. So it's a play between viability and not. And normally the extras contain less anyway.
Okay, but it sounds like perhaps there is a lot of scope there for some more research on that
definitely.
topic for sure. Great. And then someone's asking about this great question, the environmental impact of the cultivation of seaweed. So, you know, a lot of the quantities that you were talking about earlier are quite small, but if these were then to be performed on a large scale, what could you comment on the environmental impact of that cultivation?
Yeah. So in the case of cultivation, actually, the environmental impact has been described as really, really low. In fact, China is the main, those 62% that come from China, most of it is actually cultivated. Okay. Meaning there's compounds that we use in our labs. If people work in the lab in microbiology, agar, all those microbiology tests made with plates, gelatins are made with seaweed. So cultivation of seaweed comes from a long tradition in China, especially from some species. And here we're starting to do it. And the sustainability issues in the case that there's some, because there's always some whenever you cultivate, there's many researchers that have been including mixed cultivation. So cultivation of seaweed together with fish, trying to produce both and create like a mini ecosystem inside a farm. So it's really interesting field of research as well.
Brilliant. Okay, that's great. And then another question is around the protein extract. So you mentioned, for example, you know, the potential uses for protein extracts and things like mayonnaise and pasta. Is there also scope for use in high protein foods? And I suppose that might come back to the ash content that you were talking about as well. But for example, for alternatives to meat or dairy, is there scope to use some of these protein extracts in foods like that?
There's definitely a huge scope for it, especially green and red seaweeds are huge in protein content. Again, the main problem when it comes to this will be to find the proper species depending on the climate that you're working with. So Ireland, we mainly produce brown seaweed. It's basically because of our climate, and we have really good species of red seaweed as well. But red seaweeds are really expensive. So when it comes to this type of uses, it's a matter of economic value versus the materials that you have. But definitely it's a huge scope for those type of protein-rich products.
Brilliant. That's great. Thank you. Okay, so I think we have all of the questions that we've come in on the chat so far. And yeah, a lot of them there were related to the taste and things like that. So I suppose, is there anything else that you'd like to add there, Marco? Where do you see the future of this going? Are there any big things you see happening?
First, thank you so much to everybody for all the questions. They were really interesting, and I'm always open for a chat. And then I think the main topics in this area are going to come related to a few of the questions. So it needs to be multidisciplinary. You know, I'm in the food processing part, but we also have to collaborate with people from consumer perception. So we have to collaborate with people interested in developing products and how those products taste. And we have to also collaborate with the first part, which is the biomass production. We can really not have a good product if we cannot have the biomass to produce it in a sustainable way, because the wild harvest seaweed in Ireland is currently sustainable because not many people is using it. If we are designing an industry in which we're going to consume tons of that seaweed, for sure we're going to have sustainability issues. So I think a huge project englobing all these is where the whole science is going in this case.
Absolutely. Oh, that's brilliant. Well, thank you so much, Marco. And just to say that next week's talk is by Dr. Martina Wallace. And I think there's a slide that we can pop up now shortly as well. So thanks very much to everyone for joining today. It was great attendance there and great questions that came through in the chat as well. So thank you.
Thank you so much.